We are starting with slide number 30. This is an image of the structural formula for a triglyceride. The portion at the top here is the glycerol, and then what's joined are the three fatty acid tails. Okay, so a fatty acid basically is a hydrocarbon chain. See how it has carbon running through it with attached hydrogens? So this is a hydrocarbon chain with a C. When you look at a fatty acid by itself, it's got a C double bonded to an O, single bonded to an OH. But the H is removed so that the glycerol can combine with the fatty acids. But anyway, this is the glycerol is the top molecule, and then the three fatty acid tails that forms a triglyceride. And triglycerides can be solid or they can be liquid. This is a good animation to watch on fats, and it is slide number 31 if you want to write that in your notes to go back and look at slide 31 later. Triglycerides can be constructed of saturated fatty acids or unsaturated fatty acids. In saturated fatty acids, you only have single bonds between the carbon atoms, and they're going to be solid at room temperature and typically from animal sources. So butter, animal fats, these are examples of saturated fats. There are no double bonds between the carbon atoms. Let's go back to this. This is better. Okay, in between all of these carbon atoms, see this is just a single bond. Single bond, single bond, single bond. Let's look at this one. Only single bonds, only single bonds. So each of these three fatty acids are saturated fatty acids, only single bonds between carbon atoms, and it does make them solid at room temperature and typically from animal sources. And then unsaturated fatty acids will have at least one double bond between carbon atoms, at least one double bond. So these will be plant oils like olive oil. They tend to be healthier for you in your diet. Trans fats are chemically modified in the food lab, basically. They are chemically modified where they take unsaturated fatty acids and make them saturated. So that is really, really unhealthy. So they take an unsaturated fatty oil, which is a liquid at room temperature, and they chemically modify it so that it is solid at room temperature. Like peanut butter, not natural peanut butter, but regular peanut butter has trans fats in it because that's what gives it the texture, because otherwise it would have a layer of oil that sits on the top. And then we have omega-3 fatty acids, which are considered heart healthy in your diet. So foods that contain omega-3 fatty acids are good for you. In an unsaturated fat, see this one only has one fatty acid tail and one double bond between the carbon atoms. And that one double bond causes a bend in the chain, and that bendy structure makes it an oil, makes it liquid at room temperature. So even just one double bond between carbon atoms will make it an unsaturated fat. That's all you have to have for it to be unsaturated. All right, another example of a lipid is a phospholipid, and that's a modified triglyceride. So it's just a glycerol and only two fatty acids, and then a phosphorus-containing group attached. Phospholipids have a head and tail region. The head is the portion that contains the phosphate group. It is polar covalent and hydrophilic, because if it's polar, then it's soluble in water, and hydrophilic means water-loving. So the head of the phospholipid is hydrophilic, whereas the tail is a fatty acid, or the two fatty acids. They are nonpolar and hydrophobic. So in plasma membrane structure, this is what it would look like, and you can look at either picture, but you have a phosphate group attached to a glycerol, attached to two fatty acid tails. And the polar part is the glycerol, the phosphate, and then also there's a nitrogen-containing group attached to the phosphate. But this is the part, the head of the phospholipid, and this is the part that is soluble in water. So the way the phospholipids will arrange themselves when they form the plasma membrane is that the tails will face each other. The hydrophobic tails will face each other, and then the heads will face away because the heads are in contact with the fluid that surrounds the cell, which is water, So steroids are lipids that consist of 4 rings. And the most important steroid is cholesterol. Cholesterol is made by the liver, and it also can be obtained in our diet by eating cheese, eggs, and meat. But our liver does make it, and it's the starting material for the synthesis of vitamin D, for steroid hormones like testosterone and estrogen, and for bile salts, which are produced by the liver that help emulsify fats or break down fats. Cholesterol is also important in the plasma membrane. In addition to phospholipids, cholesterol is also found in the plasma membrane of our cells. This is the basic structure of a steroid. Basic structure of cholesterol is 4 rings. And the testosterone and estrogen are just modified. Will be a little change, a little modification, but it's still 4 rings. Now, our last example of lipids are eicosanoids. They're derived from a fatty acid called arachidonic acid that's found in plasma membranes or cell membranes. The most important eicosanoid are prostaglandins, and they play a role in blood clotting, controlling blood pressure, inflammation, which happens, when we have an infection, and labor contractions. Inflammatory actions are blocked by NSAIDs, which are pain medications, non-steroidal anti-inflammatory drugs such as aspirin or ibuprofen. So the NSAIDs will kind of block the action of prostaglandins. So prostaglandin is the most important eicosanoid, and eicosanoids are our last example of lipids. Now we're going to move on to proteins. Proteins make up 20 to 30% of cell mass. They have the most variety of functions of any of the molecules that we're studying. Of carbohydrates, lipids, and nucleic acids, proteins have the greatest variety of functions. There are structural proteins. There are chemical proteins such as enzymes. There are even hormones that are proteins and not steroid hormones. There are proteins for contraction that are found in our muscles, or in our muscle fibers. Proteins will contain carbon, hydrogen, oxygen, nitrogen, and sometimes sulfur and phosphorus. But definitely these 4. Definitely proteins have nitrogen, whereas most lipids and definitely carbohydrates do not. When amino acids, which are the monomers of proteins, are joined together by dehydration synthesis, the bonds between them are called peptide bonds. And when you have a chain of amino acids, it is called a peptide. Once it undergoes a lot of complex shape changes, structural changes, it will become a protein. So there are 4 levels of structure in a protein. Before we get to that, we have examples of proteins. So this is a picture of collagen, which is a structural protein. Collagen is responsible for the strength in our dermis of our skin. Collagen is responsible for the flexible strength of bones. It gives bones a kind of a flexibility and strength. The other component of bones that gives the bone its strength is the calcium. It's like a cement-like substance, And tendons and ligaments contain collagen. Enzymes enzyme proteins they catalyze chemical reactions. In this case, it's an enzyme that's catalyzing the breakdown of a disaccharide. So let's say the disaccharide is sucrose, then the name of the enzyme is sucrase. So it will kind of interact with the disaccharide and break it down, and then the enzyme can be reused. Let's see, transport proteins like hemoglobin. Hemoglobin has basically 4 globular subunits. So it's really 4 proteins that are connected, by chemical bonding. But hemoglobin is a transport protein because it transports oxygen, and also it transports a small amount of carbon dioxide through our blood. It's found in our red blood cells. We have 2 contractile proteins for movement. They're called actin and myosin. They're found in our muscle fibers or muscle cells. We have proteins that act as chemical messengers like hormones. Insulin is a hormone that is also a protein. And then we have proteins for defense called antibodies. All right, all proteins are made from 20 different types of amino acids. The amino acids are joined together by peptide bonds. And the thing that makes the 20 different amino acids different is their R group. So this is the structure of an amino acid. You have a central carbon. That carbon is bonded to a hydrogen atom, and then an amine group, which is nitrogen and 2 hydrogens. This is the amine part of it. And then the central carbon is also bonded to an acid group, COOH. That is a carboxylic acid group is what it's called. And we also have that in fatty acids as well. And the other chemical that is attached to the central carbon is the R group. And R is not a symbol for an element. R just means variable. So any chemical, any atom or group of atoms can be found here. And there are 20 different R groups, and that gives you the 20 different amino acids. All right, what we're seeing here is that when 2 amino acids are joined, it's called dehydration synthesis. A water is removed, and you have a dipeptide. Very, very similar to when we saw 2 monosaccharides joined to form a disaccharide. When the dipeptide breaks apart, it's called hydrolysis, and water is required. So you have to add a water by adding a hydrogen from the water and then an OH from the water to split that dipeptide up into 2 amino acids. That's called hydrolysis. All right, the levels of protein structure are primary, secondary, tertiary, and quaternary. The primary level of structure is just the sequence of amino acids. So just picture a chain of amino acids just lined up in a chain. That's the primary structure. The secondary structure is when that chain either forms into a helical structure resembling a spring. It's called an alpha helix. Or forms into a pleated shape, like if you were to, it says here accordion ribbons, but if you've ever made a paper fan, did pleats. All right, so the secondary structure is either an alpha helix or a beta pleated sheet. Then the tertiary structure is how the secondary structures interact to form a 3-dimensional shape. So I always use this example. In fact, do we have a picture? Yes. Okay. So there used to be these things called, we don't have a good picture. I'm sorry. There used to be these things called phone cords. You may have seen some phones that have them still. So picture the phone cord. That shape is the secondary structure. That's the alpha helix. And picture that phone cord tied up in knots, and just folded and twisted and tied up in knots. That would be the tertiary structure. Okay, so the quaternary structure is when you take 2 or more different tertiary structures and connect them. The only one we've seen so far with a quaternary structure is hemoglobin, because it had 4 subunits, 4 tertiary structures that were chemically combined. So this is the primary structure, sequence of amino acids. Secondary can be alpha helix or beta pleated sheet. Tertiary is the three-dimensional shape, and quaternary is a combination of more than one tertiary structure.